Hello everyone. So the paper that I'm going to present is complete loss of H3K9 methylation dissolves mass heterochromatin organization by Montavon et al. We know that all nucleosomes have the same architecture, about 147 base pairs of DNA wrapped around histone octamers, but histones can contain different post-translational modifications on their amino acid residues. One example is histone methylation. Methylation on lysine number 9 is a hallmark of heterochromatin, and it's important in genome integrity, chromosome segregation, and gene regulation. Histone H3 methyltransferases all contain a set domain. It has been identified in mouse cells. There are 6 genes or 3 distinct gene pairs that encode set domains containing enzymes that has methyltransferase activity towards H3K9. This paper aimed to assess the function of histone 3 methylation on lysine 9 on mouse heterochromatin structure, and then they explored differential gene expression and repeat elements in cells that are deficient in enzymes responsible for methylation. And at the end, they concluded that H3K9 methylation, specifically SUV39H, ESET, and G9A enzymes are required for maintaining heterochromatin organization in mice meth cells in vitro. To understand the importance of H3K9 methylation, they went ahead and generated multiple mutant cell lines in which they inactivated multiple H3K9 set domain genes using CRISPR-Cas9 technology in a successive way. Wild-type cells are E1C, ESET1 conditional, which is a tamoxifen-inducible conditional deletion of ESET1. They first inactivated E1 by tamoxifen, and then they transfected wild-type meth cells with plasmids carrying Cas9 and guide RNAs. So when we say E1, E2, G9, GI, this is the order of which they inactivated the genes. So they confirmed the absence of mutant gene products in the cell lines that are generated by Western blot. For example, in 6-knockout-1, they inactivated genes of all 6 methylating enzymes, so you can't probe for any of the methylating enzymes. They examined H3K9 methylation using Western blot. So as you can see, if we lose one set of enzymes, others can compensate for methylating H3K9, but if we lose all sets of enzymes, like in 6-knockout-1 and 2 mutants, we don't see any histone methylation. Findings were confirmed through mass spectrometry as well. So this confirmed their hypothesis of redundancy that exists between H3K9 methylating enzymes. They use RNA-seq to determine if various H3K9 methylating enzymes mutant cell lines show alterations in the repeat elements in mouse genomes. So they showed that deletion of ESET1 alone is sufficient to induce depression of a subset of endogenous retroviruses. And as we knock out more enzyme sets, like here and here, we see higher numbers of depressed ERV elements. They also assessed changes in chromatin accessibility by ATAC-seq. ATAC-seq revealed that disruption of H3K9 methylating enzymes lead to a global increase in chromatin accessibility. So we concluded that disruption of H3K9 methylating enzymes lead to depression of multiple families of repeat elements, and H3K9 methylation is a marker of heterochromatin. Then they analyzed changes in gene expression levels upon loss of H3K9 methylations using RNA-seq. They saw that mutant cells that were depleted of all 6 methylating enzymes show drastic deregulation of thousands of genes. And they concluded that loss of H3K9 methylating enzymes induce a signaling cascade, inducing DNA damage, sensing pathways, interferon response, and attenuation of cell proliferation and cell death. So here they visualize heterochromatin. They use DNA fish for major satellite repeats. We can see that MSR clustering is maintained in mutant cells lacking ESET1 and ESET2. But as soon as we delete S1, S2, which is the SUV39H enzymes, we see a partial dispersion of heterochromatin. So they concluded that the spatial organization of heterochromatin is mostly dependent on SUV39H enzymes, but when we knock out all of the enzymes, we see the complete collapse of heterochromatin foci in both knockout mutants. Then they further investigated the nuclear organization in mutants by visualizing the integrity of nuclear lamina. The method used was immunofluorescence detection of lamin B. They saw that mutants that lack ESET1 function, they displayed multiple macronuclei and deformation of nuclear lamina. This confirmed that E1 is an important H3K9 methylating enzyme and has a role in preserving the integrity of nuclear lamina. Then they analyzed distribution of fibrillary, which is a protein that labels nucleoli. They found that nucleoli structure was not altered in any of the cells. So to recap, there is a definitive role of H3K9 methylation in maintaining heterochromatin organization and genome integrity. Then they used TEM microscopy to analyze this collapse of heterochromatin at higher resolution. They visualized the 6 knockout cells. They found out that these cells, they lost the distinction that exists between heterochromatin and euchromatin. The white arrows shows heterochromatin. So they concluded that there are distinct functions for the SUV, ESET, and G9 sets of enzymes in establishing H3K9 methylation states. Higher degree of redundancy exists between H3K9 methylating systems, and there's a crucial requirement of H3K9 methylation in maintaining heterochromatin organization. To sum up, H3K9 methylation, specifically these enzymes, are required for maintaining heterochromatin organization in mice map cells. To criticize this paper, positive notes was that methodology used was relevant and adequate. They confirmed their results of their experiments using at least 2 techniques, and they provided a groundwork for future studies to understand the mechanism behind collapse of heterochromatin or proteomic studies to verify changes in gene expression. They provided a valuable RNA-seq data, so it will facilitate bioinformatics studies. But on a negative note, RNA-seq results could have been confirmed further by doing Western blot or flow cytometry to confirm that these changes are not only on the level of gene expression. A technical aspect that can be improved was that they didn't mention about non-targeting gRNA sequences that are negative controls for CRISPR-Cas9 technique. And they didn't say how they validated the successful genetic targeting. They said that they used Western blot to confirm absence of protein after CRISPR, but they could have used these techniques to validate the successful genetic targeting. Another thing is that they used tamoxifen to conditionally delete ESET1, but we know that tamoxifen induces epigenetic changes, such as DNA methylation in breast cancer cells. So this is a highly relevant off-target effect that was not mentioned by the authors. Thank you for your attention.